Hi everyone, welcome to our webinar. My name is Kasia Proctor and I am the Director of Marketing Communications here at Biolog. I am going to be your moderator today. Our webinar today is titled Hypoxia with the AS-500 chamber for culturing microaerophiles. First, I have a few housekeeping items to remind you of. We are recording the webinar and we will be sharing the link to the recording with everyone who had registered. If you have questions, please submit them through the Q&A chat feature. Some of you submitted questions already during the registration. We appreciate that, and we will do our best to answer them either during the slides or at the end. We do have a short survey that will pop up when you log out. We appreciate your feedback on the webinar. Now let me introduce our speaker. Max Gravener is our product manager for instruments and software. He received his PhD from University of Georgia in microbiology with a focus on fungal genetics. He worked with Candida albicans, studying natural variations among clinical isolates and how gene expression and regulatory differences can impact their ability to form biofilms on implanted medical devices. Thanks so much. Welcome, Max.
Thanks, Kasia, for the introduction, and thank you everyone for attending today's webinar. I'm really excited to talk to you about the new opportunities with hypoxia and the AS-500 chamber. To get things started, I'd like to talk a little bit about atmosphere. Atmosphere is everything. So in this example here, we have some mountain climbers going up to a high peak, and they probably have some oxygen tanks on them, as oxygen is very limited at high elevations. And oxygen is really critical for all sorts of things when it comes to microbial cell culture. Obviously, with the anaerobic chamber, we want to eliminate oxygen, but oxygen can be very important for many different applications with different types of microbes. So now I'd like to talk about the influence of oxygen on microbes and the different oxygen requirements of different classes of microbes. So oxygen levels directly impact growth and energy in a variety of different ways. So first off, oxygen is both a friend and an enemy, but this really depends on the organism and its tolerance to oxygen and its toxic products. And so different metabolic processes as well are going to influence the preference for growing in the presence of oxygen or not. Oxygen needs are different for different metabolic processes. For example, in respiration, you're using oxygen to generate lots of ATP. However, in processes like anaerobic fermentation, you're not generating very much energy and not using oxygen at all. And some organisms can deal with the toxic products that are produced by oxygen, like peroxides and things like that, in different amounts through enzymes like catalase and peroxidase, while others really can't handle any of these products, and it's actually poisonous to those strains. So next I'd like to talk a little bit about the degrees of oxygen tolerance as well as the names for these different microbial classes. And to do that, I'm going to use this pretty famous example of how to determine aerotolerance among microbes, where we have a tube of liquid media that has a culture growing in it, and we're going to look at the location of the cells within that tube to determine their oxygen tolerance. So first off, on the left here, we have our obligate anaerobes. So these first do not use oxygen for energy production, and they're also poisoned by it. They're not able to handle any of those toxic products. Next, we have our microaerophiles, and you can see these are growing not at the surface of the liquid, but down slightly, and that's because they require oxygen for energy, but they are susceptible to the toxic compounds that are produced by oxygen, so they don't want a lot of oxygen present, otherwise they can die. In the next tube here, we have our facultative anaerobes. These ones can either use oxygen for energy production or not, and the processes where they use oxygen are very energetic, and so they tend to prefer the presence of oxygen for energy production. And they're also not poisoned by those products at all. They have the enzymes to handle it, so they're able to grow throughout the entire tube, and they actually concentrate a lot here at the top. Next, we have our aerotolerant microbes. And even though these say aerotolerant, these actually don't use oxygen for energy typically. and they're not poisoned by it. So they have all those enzymes that are needed to deal with those toxic products, allowing them to grow pervasively throughout the media without any preference at the top or the bottom. And then finally, we have our obligate aerobes. And so these ones require oxygen for energy and they're not poisoned by it. And so they grow right at the top of the media where it's very oxygen rich. So today we're going to talk about microaerophiles. So these are the cells that grew not at the surface of the media, but just under it, where they're close enough to get some oxygen for energy production, but not so close as to get exposed to atmospheric oxygen and thus be poisoned. And so these strains like to grow in these low oxygen environments, which are also known as hypoxic. And what characterizes a hypoxic atmosphere is having an oxygen concentration that's typically between 1 and 7% oxygen. So traditionally, culturing microaerophiles has been done in something called a candle jar. And you may be familiar with this from either your undergraduate or research work. And this system uses a candle to consume most of the available oxygen inside the jar, leaving a more or less hypoxic environment. So we're starting out at 20.9% oxygen, or normoxia, here with our plates in a candle. And then as it burns, it's going to consume that oxygen and then get down to where the candle can no longer burn. There's not enough oxygen to sustain the flame, which is typically around 3 to 5% oxygen. And that's considered a hypoxic environment. But there's some issues with this method that make things like chambers that can maintain a hypoxic environment and let you work with your samples in that environment without disturbing them a really attractive option. And so the first problem here is going to be the inconsistent and uncontrolled oxygen levels that remain once the candle extinguishes. So I have that range here, 3 to 5%, and the reason it's not a specific number is because the candle is going to determine a lot of how much oxygen is left, how good the fuel in the candle is, the quality of the burning, etc. The size of the jar are all going to be influencing how much oxygen is left at when the candle goes out. Next, we have all the smoke and particulates that are introduced into the system when you are burning a candle here, and that can cause its own issues. And finally, when you actually want to work with these bugs, then you have to open the jar. And as I mentioned, microaerophiles, many of them can really be poisoned by atmospheric oxygen. And when you open that jar, you're suddenly flooding it with 20.9% oxygen as opposed to hypoxia, and potentially poisoning your high-value samples. And so these points really emphasize the importance of using something like a chamber to bring your entire workflow into that hypoxic environment to not risk your sample integrity. So I've talked a lot about microaerophiles and what makes them a microaerophile. And next I'd like to spend a little bit of time on where we see these hypoxic environments. And the short answer is we see them in a lot of different places. So first and foremost is going to be high altitudes. So low pressure means low oxygen. And so any organisms isolated from environments like that may benefit from hypoxic conditions. Additionally, fresh and marine water both typically have oxygen levels that are very low, so less than 1% in many cases. And that just keeps getting lower as the temperature increases. Human and animal tissue, these typically are around 3 to 10% oxygen. So any pathogens or commensal microorganisms that you isolate from the body could potentially benefit from hypoxic incubation if they're not anaerobic. Next, we have subterranean environments. So underground environments like caves will often have really poor air circulation, as well as a lot of gases like carbon monoxide or carbon dioxide, which can make the environment hypoxic. So microbes grown in any sort of these environments may benefit from hypoxic incubation or may be microaerophiles in and of themselves. And so next I'm going to go over some examples of microaerophiles. So now we're going to go over 3 examples of commonly isolated and researched microaerophiles. So the first one is going to be Helicobacter pylori. This is the most common cause of gastric ulcers in humans, so it's very important for clinical research. The next one is going to be Campylobacter jejuni. Campylobacter jejuni, along with other Campylobacter species, are going to be the most common cause of diarrheal illness in the U.S., which typically comes from consuming contaminated food like poultry. And this bug grows really well at 5% oxygen, which I'll talk more about later. Next, we have Vibrio, which you'll see come up in the news quite frequently. So vibriosis infections are typically contracted from contaminated coastal waters as well as undercooked shellfish, and those affect tens of thousands of people per year. Just to give you a few news headlines from the last year or so, you can see here we have two different Campylobacter jejuni recalls that were announced by different health administrations. And then we also have this one at the bottom here where Vibrio vulnificus bacteria was isolated in coastal waters, and they were advising people to stay away to avoid infection. So these bugs are really important for public health reasons as well as basic research. So now that I've given you a lot of background on microaerophiles and hypoxia, I'd like to talk more about the AS-500 chamber, which is a best-in-class solution for modular and ergonomic anaerobic culture with automated maintenance, and I'll be talking more about how you can add hypoxia to that list of features as well. The AS-500 chamber is really the ideal solution for anaerobic culture and handling for several reasons. So first is the automated environmental control. There's no need for daily baking of the catalyst as it's constantly heated to 100 degrees Celsius, meaning that sulfides produced by microbes, which are poison to a catalyst, never have a chance to build up on that catalyst and prevent it from catalyzing the reaction between hydrogen and oxygen. So further protecting the catalyst is going to be our anti-scrub pellets, which are responsible for scrubbing out some of that hydrogen sulfide gas, as well as the HEPA filter, which is going to take out any particulates and keep contamination risk down in the chamber. You'll also notice here that we have the AS-500 chamber on the left, and then the right half of this is actually our instrument module. And so I'll talk about this more later, but I also wanted to emphasize that the AS-500 chamber really can grow with you and your lab and your needs, and we have different modules to expand the chamber workspace to really suit your specific applications. The AS-500 also includes a vacuumless pass box with internal and external touchscreen controls, as well as a slide-out tray. And the nice part about this vacuumless pass box is that it means that there's no need to cool media before bringing it into the chamber or disassemble any bottles like squirt bottles that may leak in a typical vacuum chamber. The chamber also has pass-throughs for USB, HDMI, and Ethernet, which enables you to connect to equipment outside of the chamber. Further expanding that capability, you'll see in this top right video, there is a touchscreen monitor that we can also install in the chamber that further allows more connectivity to the outside of the chamber. Finally, the chamber is equipped with our vacuumless InstaSleeve entry, which allows you to go in and out of the chamber without having to wait for the sleeves to purge. You're also not wearing those thick, bulky gloves that reduce tactile sensation. And the added benefit of these sleeves, as you can see in the bottom video here, is that you can pass small items directly into the chamber without having to waste gas and time waiting for a pass box cycle to run. So one of the key features of the AS-500 chamber that I mentioned before is its modular expansion potential. And so we have two different modules currently for the AS-500 chamber. So the first one is going to be the expansion module, or EX, here in the middle. And this is really for adding additional space for another operator to work in the chamber at the same time. So it essentially doubles the available floor space as well as adds some more shelves for use in the chamber. Next, if you have equipment that needs to be brought into the anaerobic or hypoxic work environment, we have the instrument module over here on the left. And so it's pictured here with our Odin 8 instrument in it, which is a pretty large footprint instrument and fits comfortably inside this chamber module. And so you have quite a significant increase in the available space, both vertically and horizontally, for your instrumentation. Together with the AS-500 chamber and the expansion module and the instrument module, you really have this great system to develop a chamber that grows with you and your needs and applications. So you can see it cycling through all these different configurations. All of this is able to be done in the field. So if you purchase an AS-500 chamber now, and then next year you hire a new operator and you want to add on some space, we can do that. And then the next year, potentially, you want to bring a device into the chamber that doesn't fit, we can also do that and come out and add another instrument module on. You can also have chambers butted up against each other to add more incubator space. The configuration options really are incredibly flexible that we can do for your specific application and your needs. And they can change over time as your work changes. So I've been hinting at it for a while, but now the time has come, and I'd like to talk to you about Hypoxia and the AS-500 chamber. So introducing the Hypoxia option for the AS-500 chamber. Now this is a software-only upgrade to the AS-500 chamber, which enables you to control oxygen levels between 0.1 and 10%. And this is in addition to its existing anaerobic capabilities. You can swap back and forth between hypoxia mode and anaerobic mode quite easily without changing the gas mix. And I'll get into more details in a minute. So how does the hypoxia option work? Well, during anaerobic operation, only anaerobic trimix gas, that 5% hydrogen, 5% CO2, 90% nitrogen gas mix, is used to maintain the anaerobic environment. The hydrogen reacts with the oxygen on the catalyst and produces water, and you get that anaerobic environment. So during hypoxic operation, a mix of that anaerobic trimix gas and air is then injected into the chamber instead, which allows you to control the oxygen level in the chamber. And so one thing when it comes to injecting gas into the chamber is that during anaerobic operation, we're really just injecting gas once in a while to maintain pressure in the system. But now with hypoxia, the gas injections will occur periodically to maintain or change your oxygen levels depending on what you need. Now I'd like to talk about how to get the hypoxia option. So we have a couple of different scenarios here. The first one is that for those of you who need hypoxia immediately when you're purchasing your AS-500 chamber, perhaps you're just purchasing it for hypoxia and don't even need the anaerobic functionality. Well, in those cases, the hypoxia option can be enabled at the factory. So whenever you receive the chamber, there is no need for you to do any further setup. You just turn on the chamber with the gas and you'll be off to the races in hypoxia mode. If you want an AS-500 now, but you're not quite sure that you need hypoxia, or you know you'll need hypoxia, but maybe down the line and need to apply for some additional funding, we also have you covered there. The hypoxia option can be activated at any time on your AS-500. And then finally, if you have an AS-500 right now today and you want to add hypoxia, we also have you covered there. The hypoxia option is compatible with all AS-500s that are currently out there in the field, and all that requires is a simple software update. So how do you actually use the hypoxia option once it's activated on your chamber? Well, it's really easy, just a couple of steps. So first off, you just need to turn on hypoxia mode in the settings, and then you'll set your oxygen level. And then the last step is to just walk away and let the chamber do the rest. It'll naturally come up to your desired oxygen level over the course of a couple of hours. No switching gas tanks, no hardware changes required. Just set it and the chamber will do the rest. So now that I've talked about the hypoxia option for the AS-500 and how it can be installed and how you use it, I'd like to talk about Odin and using Odin in the AS-500 with hypoxia to characterize some microaerophiles. So in addition to chambers, Odin is our flagship instrument that is a real workhorse when it comes to microbial characterization as well as identification. And here in this application, we're using Odin to characterize microaerophiles inside the hypoxic working environment using the AS-500 equipped with an instrument module. And so in this context, we're using Odin 8. And so Odin 8, it reads and incubates up to 8 microplates at a time, as opposed to its big brother, the Odin L, which can do up to 50 plates. The Odin 8 fits nicely in the anaerobic chamber instrument module. And so during use with the Odin, you're able to monitor growth curves in real time, and you can use that data then to analyze either single isolates or communities, and then compare those samples with the Odin analysis software to get really actionable results. So our lines of phenotypic characterization plates really make high throughput screening efficient and easy. And things that you can do here include comparing substrate utilization among different candidates or communities. You can also use the plates to spot key sensitivities as well as condition requirements by screening your organisms in a variety of different conditions. So the characterization platform I want to focus on today with this application for microaerophiles is going to be our phenotype microarray, or PM plate series. And so this is a series of plates where you can screen for substrate utilization, pH, salt, drug sensitivities, and all sorts of conditions. And each plate is going to have a different condition in each well. So I'm going to focus on 2 specific groups of plates. So first off is PM1 and PM2. And so these are our carbon source utilization screening plates. Between the 2 of them, they contain 190 unique carbon sources, which probe a variety of different pathways. The second group of plates I'm going to talk about are PM11 and PM12. And so these are 2 of our antimicrobial dilution series plates. And so each of these plates are going to contain 24 antimicrobial compounds in dilution series. And so between the 2, we have 48 compounds total. Finally, for hypoxic PM, we're going to be measuring growth over time. So now I want to go over a little bit of what this data typically looks like. And so here's a sample of what we call our curation view. This allows you to look at replicates side by side. So here we have 2 replicates for wild type, 2 replicates for mutant. This is not hypoxia data. This is just some general data to familiarize you with what this looks like. And so we can see here for these 2 compounds that the replicates are quite stable for both the wild type and the mutant, and you can see some pretty clear differences. This is a really nice tool to allow you to do some quality control on your data before moving forward in analysis. Speaking of which, as you move forward in the workflow, you'll then be able to aggregate those replicates together and then calculate useful parameters and the difference in those parameters between your reference and your test. So in the example here, we're looking at the wild type as our reference in pink and the mutant as our test in cyan, and calculating the difference in the area under the curve between those 2 samples. And you can see here that those 2 compounds, again, the mutant has some significantly increased resistance to those. And I'll tell you they're tetracycline compounds, which makes sense for this mutant as it has a tetracycline resistance marker input into its genome. So today I'm going to be focusing on Campylobacter jejuni. So Campylobacter overall, as I mentioned before, is one of the most common causes of food poisoning. So the CDC estimates that there is about 1.5 million cases per year, making this a really important bug for study. And where we find it is mostly in poultry, but it can also be transmitted through other modes of contamination as well. So typically, Campylobacter is happiest at about 5% oxygen, but it can persist outside of those conditions, such as those in food packaging, for weeks at a time. Finally, one of the things that's known about Campylobacter is that it undergoes phenotypic changes when you change the oxygen level. And so that's the thing we're going to be probing here in this study with Odin. So let's take a look at the data that we actually generated with Odin and Campylobacter jejuni in the AS-500 enabled with the hypoxia option. So the first data set that I'm going to show you is with PM plates 1 and 2. These are our carbon source utilization screening plates that contain those 190 different carbon sources. And so we did this assay at 37 degrees at 5 or 10% oxygen. And so figures I'm about to show you will have pink for 5% oxygen, cyan for 10% oxygen, and then this blue color for their overlap. So here are 3 of the carbon substrates that showed some differences between 5% and 10% oxygen. So we have beta-methyl D-glucoside, succinic acid, and D-L-malic acid. You can see here we have OD at 590 nanometers going up the y-axis, and then we have our time on the x-axis. So what we can see here is that carbon source preferences do absolutely change depending on the oxygen level. So for instance, the C. jejuni strain we used here grew at 5% much better using beta-methyl D-glucoside rather than when it was growing at 10%, using that as its sole carbon source. Then if we take a look at the bottom 2, these organic acids, the reverse is actually true, where the 10% oxygen sample grew much better, much earlier than the 5% oxygen sample. Interestingly, with succinic acid, 10% oxygen yielded a higher overall OD than 5% oxygen, but with D-malic acid, both 5% and 10% reached the same level, but at 10% oxygen, growth picked up much sooner during the growth period. So this just shows how carbon source utilization can change with oxygen level. So now I want to talk about Campylobacter with drug sensitivity screening. So we looked at PM11 and PM12. These are 2 of our drug screening plates. Again, in the same conditions, but this time we're in rich media instead of in nutrient-limited media, looking at substrate utilization. So if we take a look at these 2 sets of figures, on the top we have carbonicillin, on the bottom nafcillin, and the concentration of these antibiotics goes up as you move from left to right. And so there's a couple of trends that we can identify here. So resistance to these compounds seems to increase with oxygen level. And so if you take a look at the left-hand side here of this figure, for both carbonicillin and nafcillin, the pink line, that 5% oxygen experiment, that reached a higher overall OD. So at low drug concentration, meaning the least restrictive, the bug seems to be happier at 5% oxygen. But interestingly, as we increase the drug concentration, the 10% oxygen sample actually does better than the 5% oxygen sample over time. So if you take a look at these cyan curves and where the overlap is, you'll notice that the 10% oxygen growth curve doesn't really change at all for carbonicillin as you move from low concentration to high concentration, as opposed to the 5% oxygen where it tapers off as you go up. And similarly with nafcillin, you have good growth at 5 and 10% oxygen in that first low concentration, but then from the second concentration well on, there's virtually no growth at 5% oxygen, but you still get growth at 10% oxygen. So this just kind of drives home the importance of testing these microbes at different oxygen concentrations and how having that flexibility in a chamber like the AS-500 can give you some really valuable and actionable results. So throughout this talk, I've gone over quite a lot when it comes to microaerophiles and the anaerobic chamber, and I hope that it's gotten across how Biolog can really help control oxygen in your research and in your lab. And that precise oxygen control is really a valuable tool in your toolkit for managing these microaerophiles, as well as anaerobic and facultative anaerobic strains. So we recognize here that complex workflows require flexible configurations. So your workflows are all going to be very different. And depending on what you're doing, you're going to need different equipment. You might need multiple users. You might have different temperatures that you will need for incubation. And all of that requires a flexible modular configurable system, which we have here with the AS-500, the expansion module, the equipment or the instrument module, and the hypoxia option. Precise oxygen control really helps to accommodate a wide range of organisms, everything from the most obligate of anaerobes all the way to microaerophiles requiring up to 10% oxygen in their atmosphere. And like I said, these flexible configurations really just mean a custom tailored chamber for you and your needs. And what you're doing may change over time, and your chamber needs to change with you. And as I mentioned before, you're able to pick and choose the modules and of the AS-500 chamber that you want to put together and change those over time as your lab grows or shrinks or your research needs change. Just as a reminder on the configuration options that we have available, first off, we have the expansion and instrument modules for increasing the amount of space in the AS-500 chamber. The EX is going to give you that extra working space, whereas the instrument module, or IM or IMO, are going to give you even more space that's good for larger instrumentation that needs to be in that controlled environment. We also have the hypoxia option we talked about a lot today for that precise oxygen control from 0.1 to 10%. And then finally, for ergonomic setup in your lab, we have our fixed height benches at desk height or counter height, that 29-inch or 36-inch for our modular configurations. And then if you're just getting an AS-500 by itself, we also have some hydraulic benches available just for the standalone chamber. And so altogether, these options present a really flexible solution for whatever your workflow may need. So I wanted to go over a little bit about service, support, and training. All chamber installations and services performed by Biolog or one of our authorized distributors if you're outside the US or Canada. And we also offer one of the only 2.5-day hands-on lab-based training courses out there for anaerobes at our Morgan Hill, California location. And at that course, it's a really valuable learning opportunity. And in the classroom portion, you'll be able to learn all about oxygen toxicity, specimen collection and transport, how to pick the right media for culturing your finicky anaerobes, as well as how to grow and isolate and identify unknown organisms. And finally, you'll get to also learn about how to manage microbiome samples without losing that community diversity that's so important. In the laboratory portion, you'll get a lot of direct hands-on experience in the chamber with culturing organisms in real-world scenarios. You'll be able to take an unknown sample of a mixed culture, pick out your unknowns, isolate them, and then get a presumptive identification using some differential media. And then finally, you'll get a really in-depth opportunity to go through chamber operation, maintenance, and troubleshooting, things that we just can't have time to cover during the in-person installation training, just in case you need to do some higher level troubleshooting. To round things out, I just wanted to touch on briefly our whole ecosystem of products and services. So for instruments and software, you've heard me talk a lot today about characterizing things with Odin 8. It also has a larger companion, which is Odin L, that can run 50 plates at a time, as well as the ID station, which is our single plate identification system. The AS-500 chamber is not our only anaerobic chamber, though it is the only one that's compatible with the hypoxia option. We also have a smaller one called the AS-150 that we'd be happy to talk to you about. For consumables and media, we have a variety of microplates compatible with the Odin system that do identification as well as phenotypic characterization and community analysis. In addition to those, we have a variety of culture media for anaerobes as well, both in the form of solid media and liquid media, in addition to some transport media. And we also offer the ability to do customized microplates and culture media as well. Our fully certified lab services lab is able to perform all sorts of identification and phenotyping services if you're looking to outsource those types of things as well. That about wraps things up, so thank you all very much for tuning in. I hope that today's presentation was insightful and helped you to understand how using the hypoxia option with the modular AS-500 chamber can give you actionable results as it grows and changes with you and your lab. We'll start taking some questions from the chat, but I also wanted to ask that you just take a couple minutes after the webinar to answer a few questions in our survey. Thank you again, and please do not hesitate to reach out for more information at info@biolog.com. Now we'll move on to our Q&A.